Okay, I want to welcome Emil Lou to give this morning's seminar. Emil is a member, a steadfast member of the Tumor Microenvironment program, and you'll see why in a minute, because it's a microenvironment kind of thing. Emil studies tunneling nanotubes, and these are structures which, in my view, are kind of on the cutting edge of how do cells communicate to one another. You've heard of soluble factors and exosomes. These are actual direct connections between cells, and they seem to be related to the malignant phenotype of tumors and perhaps can transfer important information, for example, resistance and so on. So I think we're in for a very interesting talk. Emil is an assistant professor, HEMOC, and he is extremely active in research. He has several honors that he's received. He has been funded by a KL2, which is a terrific accomplishment here at the U, and he is also has other local funding to get these things moving forward. And I'm really looking forward to having him talk. I was just talking a lot with him about what might they be, so hopefully we'll hear more about what might they be. Emil, thank you so much for talking.
Thank you, Jim. Thank you so much for the invitation and the opportunity to come speak with you today. I've been here almost, this fall makes 5 years, so I thought I would use the opportunity to also just introduce a little bit about myself and also use it as an opportunity, not just as an introduction, but maybe as an opportunity, a jumping board to facilitate whether it be potential discussions, collaborations, or further. As a both clinician and a scientist, I find myself straddling both worlds, and I think like many clinician investigators or whatever we end up being called, it's an ever-rising challenge. But I think in this era of team science, it's really important. So I thought it'd be, just so you understand where I'm going with this, especially in the first half of my talk, I wanted to share a little bit about my philosophy on and the background that shaped how I had this perspective. I did a combined MD-PhD program, but when I did that, it was the paradigm was completely different. We heard a lot about bench to bedside. So that presumed that the findings in the laboratory were going to be relevant clinically. But then I emerged 2 decades later, practically after residency and multiple fellowships and all the training, to find that the paradigm was quite different. And I'll give you firm examples, both here in our own lab and also just a lot of what I observed during my own training of observations whether literally at the bedside or in the clinic, that informed how the research would be geared, and a lot of those are in clinical play and have become the standard of care just in the last 5 to 10 years. At Sloan Kettering, as an oncology fellow, I focused mostly with the lung group, and I also did a lot of GI oncology. So that, of course, would encompass pancreatic cancer, metastatic colorectal cancer, metastatic lung cancer, among the worst of the worst. And to compound that, I did an additional fellowship in neuro-oncology, which I think it doesn't need to be clarified any further. These are just vicious tumors that are often incurable or untreatable. Here in my clinical practice at the university, I focus on gastrointestinal oncology, especially pancreatic and colorectal. And the common theme that's woven between what I'm doing clinically and what we're aiming to do and our goals in the lab is to really take a fluid bench-to-bedside model in which there isn't one precedent over another, but constantly going back and forth and really looking at investigating what cellular or molecular mechanisms are not known and what we can do to investigate them in the lab, especially for these drug-resistant cancers. And at the end of the day, I think what drives me, and I think what really drives team science, what needs to drive us as researchers, is understanding why we do this. It's not for, we'd all love high-impact papers and first authorship papers and grants, of course, to drive our passion science, but we do this with the eye towards curing the human condition. That's really the goal. Okay. In 1971, President Nixon declared what was known as the War on Cancer. He wrote the National Cancer Act, and the goal was to cure cancer within 10 years. And it's only with the hindsight of history that we might think that was either foolish or incredibly naive. But it's important to keep that perspective because it's very relevant to today. When I was at Sloan Kettering, the seminar room where there were often basic science seminars and tumor boards alike, very appropriately, this plaque was in the back, and it was the pen that was given to Benno Schmidt as he signed the National Cancer Act. He was a venture capitalist in New York City who was appointed by Richard Nixon as chairman of the President's Board until 1980. So that 10-year span came and went, and what we didn't have at that time in society were a lot of the molecular and cellular technologies. So basically the stuff that you do in the lab today that would have either taken years to do what you can do in days now, let alone genomic sequencing. But really, a lot of the things that we do commonplace were not available. But the perspective also was missing. Richard Nixon said in the State of the Union address in 1971, "I will also ask for an appropriation of an extra $100 million to launch an intensive campaign to find a cure for cancer. And I will ask later for whatever additional funds can effectively be used. The time has come in America when the same kind of concentrated effort that split the atom and took man to the moon should be turned toward conquering this dread disease," treating it like a singular disease, which we know now is not the truth. "Let us make a total national commitment to achieve this goal." This is very relevant because just a few weeks ago, Vice President Biden came to the ACR annual meeting and talked more about what we know as a Cancer Moonshot Initiative. And this is what we're all striving for, right? Whether we're in the lab as basic scientists, whether we're translational researchers, or in the clinic treating these patients. But the question that weighs on my mind, and rather heavily, and thinking how it would weigh on what I do as a researcher, is questioning, will it achieve its aims, or are we going to make the same mistakes that we made the first time? Just to give you a full disclosure, I wasn't even born when the National Cancer Act was put into play in the war on cancer. So I wonder, putting myself at this podium 40 years from now, if I'm still alive, I hope you're invited, but maybe a young physician scientist who's not even born yet today will be here talking about the Cancer Moonshot Initiative. And I wonder what he or she will be saying. Will they talk about the successes that we learned the second time around? Or will they be talking about how history really repeated itself? What's going to be different among us? What's going to be different is not just the technology that we have in the lab, but are we going to acknowledge failures that we've propagated in society and in the world of science, in the world of medicine? Are we going to reassess our approach this time? Are we going to understand cancer? It's not that we can't, but it's really a question of, are we willing to do this? We need new strategies. We have plenty of new tactics. So I'll talk more about that analogy. Tactics are the armamentarium that I, as a clinician, can use. New drugs that didn't exist even when I was in fellowship or residency. That's not the problem. It's how to use them. It's the strategies, both in how we approach our research, whether we're talking about cells or molecular pathways and mechanisms of resistance, or when I'm faced with a two-dimensional scan of a patient whose pancreatic tumor grows, I'm making a very rudimentary decision of something based on something that's just not ready for three-dimensional impact. We're just not there yet in many ways, but we can be. That's the goal of the Cancer Moonshot Initiative. Combining cancer genomics and, as Jim alluded to, the tumor microenvironment. These are, whether we study a tumor microenvironment or the genomics or whatever we focus on, it's important to understand, just as we treat patients as a whole patient, the whole tumor, the whole patient that harbors the tumor that spins off to become metastases. We're very naive in many ways clinically, and we're so far behind what we know about the research. And that's where a lot of what we have to catch up in the era of translational science. How do you account for a constantly moving target? We treat patients, even in this era of targeted therapy, with the targets that were present that we tested at the time of diagnosis. And then they spin off metastatic tumors, and we presume, very naively, that the tumors that are in the metastatic sites will be susceptible to the targets that we identified in the primary tumor. And we know that not to be true, yet we're not there in the clinic. We're just not doing it. Strategy and tactics are really important. And other fields in society use it. Why can't we? So Sun Tzu was an ancient Chinese military strategist thousands of years ago. His book, The Art of War, is one of the 7 military greats of Chinese culture. This is The Art of War is as much mandatory reading at West Point as it is the Wharton School of Business. I take inspiration from this. I have a copy on my shelf, and when I'm sitting there stuck on a grant or I don't even know what direction I'm supposed to take, I take it down from my shelf and I flip through it, and I'm going to share some quotes with you today. And in my mind, I'll tell you how some of them relate very well, or very pertinent to things, I think, clinically or scientifically. To know your enemy, you must become your enemy. That sounds very harsh and a little unusual. But we have to think like cancer thinks. We have to think, if I were in a position where I were being bombarded with someone trying to oppose me, how would I get around that defense? If you're on a basketball court and you want to take a shot, you can make a lot of shots unopposed. The moment a defender comes, the moment you're double teamed, for a while you might sputter, but then you can find ways around the defense. Cancer does that and more. It is said that, and Sun Tzu said, it is said that if you know your enemies and know yourself, you will not be imperiled in 100 battles. If you do not know your enemies, but do know yourself, you will win one and lose one. basically you break even. That's not what we're going for in human patients. If you do not know your enemies nor yourself, you'll be imperiled in every single battle. You lose. So the correlates to that, in my mind, and I put a question mark after this for the first, if you know the cancer that you're dealing with and you know that the armamentarium that you have at your disposal, if we use the strategy and the tactics correctly, will this be the new paradigm for molecular and cellular oncology in the 21st century? Just in the last decade, we're on the road, but I think with the hindsight of history, 50 years or 100 years from now, long after we're gone, this will be the dawn of that era. I think that's very clear. But if you break even, that to me is like having information at your disposal but not using it correctly. And one of the things that comes to mind is validated biomarkers, whether it be KRAS or EGFR receptor. In many cases, we have them or we're not using them correctly. If you fail miserably, I think of that as 20th century oncology. Chemotherapy, in many ways, I think the goal is to make it obsolete. Right? So it's not that it's not targeted, but it's not selective. So if I give these chemotherapeutic drugs that have been around for half a century or many decades, they are targeted. So we almost falsely talk about targeted therapy as being a new thing in the last 5 to 10 years. They're not selective. So that's why patients still have a lot of side effects from them, and that's why eventually these cancers become resistant. So the model of bench to bedside being fluid, you can think of it as linear or you can think of it as being circular. It's fluid. So there are different kind of categories. A lot of what maybe people in this room or a lot of what else do is maybe in T0 to T1, we're at the bench, we're talking about cells and molecules and molecular pathways and animal models, and it goes all the way to the other end of the spectrum. So I'll give you an example of something that is very real today in the clinic that wasn't 5 to 7 years ago. With the propagation of tyrosine kinase inhibitors that were brought to clinical trials eventually throughout North America, Europe, and Asia, there was a landmark trial in non-small cell lung cancer called IPAS. And one of the oncologists that was in the group that was at Sloan Kettering was a T4 or T3 researcher. And he looked at the data from that and his own trials, and they could have said the drug just doesn't work, gefitinib, or what's known as Iressa, which is now FDA approved for that cancer. But they did a subset analysis, which has really become en vogue. And they said, you know what's really unusual? This is a smoker's disease. I put that in quotes. It's a smoker's disease. But non-smokers who are women, who are young and Asian, for whatever reason, that subset is getting lung cancer, but they responded. The others did not as well. But statistically significant response was seen in that subset. So then they went back and had this discussion. So this is really, to me, is a great paradigm of the importance of communication in translational research. And in the room, there were also basic scientists and translational research, one of my role models, Will Powell, who was also seeing patients, and he had a whole lab, and he worked under Harold Varmus. So they went back to the lab, and at the same time, 2 other labs in America and also overseas identified the epidermal growth factor receptor. And so the response rate, just to give you a perspective, chemotherapy and that cancer response rate, is it going to work or not, 15 to 20% with chemotherapy, was as high as 70% with the tyrosine kinase inhibitor. It's not 100%, but that quickly entered from only a few places testing for it to by end of my fellowship, a lot of places testing for it, to the point where it's malpractice if you don't test for it in a new patient with metastatic non-small cell lung cancer in 2016. And then we've also realized it's not the end-all and be-all of targeted therapy. Because as we've realized as years have gone by, celebration subsided, you realize these patients progressed. And when the patients progressed, we need to find out what molecular mechanisms of the EGF receptor led them to no longer respond to those tyrosine kinase inhibitors. So now we have 2nd generation and 3rd generation. We're going to have more. But understanding that really required that bench to bedside back and forth discussion. I'll give you just a couple examples among my translational research team. I had a patient with what I call an exceptional non-response. In the last 5 years, there's been a lot of whole exome sequencing and understanding and plucking patients from clinical trials that were deemed to be ineffective, but then identifying patients who are called exceptional responders. Their tumors had exceptional response to the target therapies in these trials. And there's been a lot of information gleaned, new targets seen, even if it's a small subset of those patients. I propose, and my team is working on what I call the opposite end of the spectrum, exceptional non-response, or chemorefractory tumors. I had a patient with basically a widely metastatic colorectal cancer that just really did not respond very well at all to any of the chemotherapies that were standard of care that we threw at him. And when I linked and talked to our molecular pathology team, we identified a novel NRAS variant. And so it had only been even identified in just one small case report overseas, and we're doing whole exome sequencing. And part of another level of communication is I had a good relationship with the patient and his parents. He did not have a spouse, but they actually wanted to will their body to science. And they said, do you think that's possible? I said, well, it's definitely possible, and I have an idea. So with his consent and full blessing of the family, we did autopsy at the time of his death, and we harvested. There were over 100 metastases. But we harvested tumors and normal tissue from every site. We're doing whole exome sequencing. And this is a great collaboration example with molecular pathology, Andrew Nelson, Subramanian, and Sarver from the Department of Surgery. And we're working also at the national level with the NIH and NCI RAS Initiative. The RAS Initiative was launched by one of the world leaders in RAS, Frank McCormick, and has a robust team of bioinformaticians, people at the bench, people in the clinic, translational researchers, who are dedicated to making RAS the untargetable target to make it targetable with drugs or other approaches. So about 30% of human cancers are impacted by mutations of RAS. There's a whole bevy of different ways to approach it. So they've been really great in collaborating with us. There are a lot of things, examples of things that we do that require people to speak different languages. We talk about translational science a lot, but it also is important to know what languages you speak and what you don't speak, and get people who speak the other languages and who know the common languages to go towards a common goal. That's really the crux of what team science has become. Another example of what we're doing is I had a patient, an adult patient with unresectable medulloblastoma, which can be really a horrific and invasive tumor of the cerebellum. There are only about 400 cases per year in adults. So this patient had an ECOG score of about 3. He had a Karnofsky performance score of about 40. And if you want me to interpret that and translate that for you, this is a guy who was wheelchair-bound. He could not get out of his wheelchair. He had intractable nausea. There was no way he could do anything like toilet for himself, feed himself, or in any way have any capacity. The standard of care is extremely limited because there have been no clinical trials, and there probably never will be in adults because the population is so limited. So it's borrowed from the pediatric experience from the late 90s, the 20th century protocol from children. Standard of care is to do surgery if can be attempted, and it could not because it was unresectable, multifocal, and radiation to the whole spine and to the tumor. And this guy, I think chemotherapy would have killed him. But I delved into the literature and found that a high preponderance of tumors in adults had upregulation of the Masonic hedgehog pathway. So knowing I would not give him chemotherapy, trying to be a responsible clinician, it just so happens that that same year, a hedgehog inhibitor called vismodegib, or Aravig, had just been approved by the FDA for basal cell carcinoma patients. It also expresses the hedgehog pathway. And I gave it to him, and within 6 months, the tumors disappeared. I saw him on Friday. This guy can lap around me now. So this was the other end of the spectrum, exceptional response. Because we did not have enough tumor to do whole exome sequencing, we did another novel thing, a novel technology that's come out in the last year or two, and did liquid biopsy with circulating free DNA. Identified a novel, not a novel, but a very rare p53 mutation associated with worse prognosis in patients with hedgehog pathway expression of medulloblastoma. And we're doing whatever we can with whatever remaining tissue to do sequencing and understanding about the Masonic hedgehog pathway. My KL2 project also is something that arose out of T1 work in the lab. I have the nanotubes, but looked at microRNAs that are upregulated and looking at differences between platinum-resistant, chemo-resistant ovarian cancers. Those are platinum-sensitive, and also looking at circulating tumor cell enumeration. So this project was an outgrowth and translational arm of what we were doing at the very basic cellular level. So Sun Tzu said, "If I know our troops can attack, but do not know the enemy cannot be attacked, it is only halfway to the victory." So for that, what I think of is if we don't know what's going on, I think of patients that I see who come when they're already diagnosed and for whom there are no screening tests for those cancers. So carcinogenesis in those early stages is an insidious process. It comes up, and we are behind before we even get started in the clinic. These pancreatic tumors, there's a reason why 85% are unresectable. If it's unresectable, there's no chance of cure. Of the ones that are potentially resectable, there's a high preponderance of those to come back even with chemotherapy in the postoperative setting. We're behind before we even get started. We don't know our enemy is attacked, and we're getting in on the war after it's already started. If I know the enemy can be attacked, but do not realize our troops cannot attack, it is only halfway to victory. So I think one of the biggest mistakes we make in oncology and in cancer research is missing the timing of treatment. We've identified the tumor, and then sometimes we do resection, whether it be of really invasive forms of breast cancer or pancreatic tumor, and then we wait. We just wait. We wait. So we know that there are microscopic cells left behind, glioblastoma. We know 100% of the time it's going to come back, and we wait. And our excuse has been wound healing or let the patient recover, but then are we winning that small battle, and we're ultimately going to lose the war because in that perioperative period, we're not treating the very clones that are going to arise and emerge. By the time we come back with our rudimentary chemotherapy, they're not going to have any effect, whereas maybe they would have had an effect at the time before and leading up to and immediately following surgery. The only trial that I know about personally is using that approach is perioperative metformin, actually, to shut down the mTOR pathway. It's an indirect mTOR inhibitor. It was done at Princess Margaret Hospital in Toronto for breast cancer. I don't think the final results, to my knowledge, have been released, but that's, to me, was a novel strategy using armamentarium that's available. I did training in neuro-oncology, so I'm one of about 5 physicians in the country that are board certified in both medical oncology and neuro-oncology. So I think of glioblastoma as just the paradigm of badness. It's a malignant tumor. It's awful 5-year survival, and we've made minimal incremental inroads in the last few years. I don't think it's an exaggeration to say the field of neuro-oncology lags at least 10 years, if not 15 years or more, behind the rest of molecular oncology. This was a patient who was very robust gentleman who succumbed to what, if it looks awful, it was awful, an unresectable form of GBM. But it is also a paradigm, at least what we've done, what people have done in the lab, drifting away in numerous types of cancers, gastric cancer and GBM, away from purely identifying them by their histopathologic subtype, but reclassifying them with molecular subtypes. So identifying classical, which is really bad with EGFR pathway or PTEN, mesenchymal, which is probably the worst subtype of prognostic-wise of glioblastoma, recognizing there are multiple types. But what do we do in the clinic? If there's a patient who's newly diagnosed, if at best they have a gross total resection and they undergo standard of care chemotherapy radiation, all we're throwing at them is what? An alkylating agent, temozolomide, that's been around for 10, 15 years. We're not even close to even addressing this. No one's even asking the question yet, clinically, mainstream, what should we be doing for classical versus mesenchymal versus neural? We're so far behind. There have been some really elegant studies as people, bioinformaticians and mathematical oncologists, and you heard from Jasmine Fu a few months ago, who I think is doing fascinating work with Kevin Leder. Intratumoral heterogeneity. If this doesn't blow your mind, I don't know what will. It blew my mind that in this PNS paper, it identified 10 patients, 6 of whom had multiple subtypes within their individual tumors. How do we address that? How do we address that? When within those tumors, there were some clones that were involved in neuronal degeneration or generation. Others were responsible for tumor angiogenesis. The cancer is using multiple strategies and tactics to attack, and we're not even up at that level. Further, in this paper, they took from this patient, this is a resection cavity, and this is blown up. So this is not the brain. This is actually the resection cavity. But look at the actual subclones, and they did a phylogenetic tree to identify the founding clone and all the spin-off clones. This is so far removed from what we do in the clinic yet. There are a lot of select targeted therapies in development. There's hope that these will pan out, whether it's one that will work and we'll use them sequentially. Should the main method of attack be sequential, or should it be at the same time? Remains to be borne out. But understanding that you can't do clinical trials for everything. You can't try every single strategy and tactic in clinical trials because clinical trials take millions and millions of dollars. So we really have to be very careful as we marshal our resources. When we take something from the lab and bring it to the clinic, A, is it safe for human patients? But if you could only do a trial once or a couple of times, what's the right way to do it? What's the rational way to do it? You'll never know until the trial is done. But putting that forethought on with a different perspective can really make a big difference. This sonic hedgehog pathway inhibitor is the one that I use for that glioblastoma patient. So if you think of the two-dimensional rendering of what we see in the lab of a three-dimensional occurrence, I think of tumor recurrence following resection. This is just a rudimentary scratch assay. It's just two-dimensional. But in my mind, I'm looking at it, I'm thinking, what would that, in what way, if that could represent tumor recurrence? This is just a toy from my toddler son, But when someone showed it to me, I said, I must show this in PowerPoints because I look at that and I say, well, that's three-dimensional. A lot of what we do in tumor boards, I go to tumor board and a pathologist is there and they show you this nice thin cut slice that's pretty in pink. And it's two-dimensional, and we just think of cancer as just a sheet of cancer cells. And it's so far from the truth. It's this. And cancer isn't just cancer. It's stromal cells. It's the tumor microenvironment. It's the vascular endothelial cells that are creating angiogenesis and disrupting the tumor and feeding it despite the chemotherapy that we're giving it. It's the fibroblasts that are feeding and soliciting and really pushing, egging on the cancer cells. And it's immune infiltration cells that might be worsening the tumor as well. Sun Tzu said, if your enemy is secure at all points, be prepared for him. If he is in superior strength, evade him. If your opponent is temperamental, seek to irritate him. Pretend to be weak that he may grow arrogant. If he's taking his ease, give him no rest. So we are fully immersed in this era of immunotherapy. It's been the hottest topic in the news and in the ACR meetings and ASCO the last couple of years. If you hear Jed Wolchok, who is now one of the international leaders in immunotherapy, it's not like he started working on this 5 years ago, but he was one of my program directors at Sloan Kettering and a consummate physician scientist and great role model. And if you hear him tell the story when they did ipilimumab, the CTLA-4 inhibitor, it's an interesting story because what we have criteria for how we interpret CT scans. Does it grow? It means the therapy is not working. Does it shrink? It means the tumor therapy is working. So he had a patient with widely metastatic melanoma on trial. The ipilimumab was given. 1 month later, there was just gigantic. If you just measure it, it was growth. So do you take the patient off the trial, or what do you do? He goes into the room. The patient says, before you tell me what the results are, I want to let you know I'm feeling better than I have in years. Ever since I was diagnosed with this, I have not felt better. So Dr. Wolchok is thinking, well, okay, now what do I do? So he shared the results with the patient, and they came up with the idea, let's give it 1 more month. Because immunotherapy may not work or may not be measurable the same way we think of chemotherapy being measured. So lo and behold, a month later, there was a severe retraction of the tumor. And so now we know, we call it a flare. It was just a flare by the immense immune response that was marshaled by the CTLA-4 inhibitor. But if you measured it, if you looked at that with the same eyes and perspective pre-2010, it very easily could have gone the other way. A major breakthrough in cancer therapy in general and immunotherapy could well have not come about if we had thought of it with the same old perspective. If his forces are united, separate them. If sovereign and subject are in accord, put division between them. Attack him where he's unprepared. Appear where you are not expected. So it goes back to this paradigm of perioperative. So you take out the bulk of the tumor. Residual cells are left behind. They're scattered. They're not yet communicating with each other. The microenvironment has not yet reformed. The subclones that might want to declare themselves as being the winners of the new tumor maybe have not yet been able to take hold. But we're not treating it at that time. We wait 4, 6 weeks, 8 weeks. And my fear is that the standard of care is just not working for that reason. So the paradigm is development, emergence of chemoresistance. But we're also learning more, for example, in colorectal cancer. There was a great study out of New York, another one in Italy, 2 published in Nature in the same year, in the same issue, that showed that the subclones of KRAS wild-type tumors, metastatic colorectal cancer, were actually harboring mutant subclones at the time of diagnosis. They didn't necessarily emerge over time. They were already there. So if they're already there, should we be coming up with KRAS-targeted therapy? And if we had it, first of all, could we use it up front, or should we be using it up front? So I think Jim is marveling that I haven't said the word nanotube significantly yet. So overarching themes. This is a bonus. I'm glad you see it that way. In terms of what we look at, that's a long background, but if we look at our strategy, and we look at our tactics in the era of the cancer moonshot, how are we not going to fail? Not to say that the war on cancer entirely failed, but it's not even close to what we thought it would be 40 years ago, or what people thought at that time. We've made a lot of developments in the technology realm, but we also have to change our thinking. We're investigating the development of intra and intratumoral heterogeneity, things that we have to at least think about, how drug and chemo resistance emerges, or if it's already there, how we encounter it. We're looking at the cellular mechanisms, molecular targets driving mutations. They can't be divorced from each other. They're not separated. We have to at least acknowledge them in tandem. What are biomarkers that either drive or are passengers along for the ride? Using technology that we use in the lab, like microscopy, is looking at tumors in a 3-dimensional manner and trying to go back to the lab and say, these are how they exist three-dimensionally. How can we attack them and understand more about them? Is cell-to-cell communication the missing link? So malignant tumors we know are highly heterogeneous entities. Intratumoral heterogeneity is something that we take as common now, and maybe it wasn't as common to talk about it 5 to 10 years ago. I can pretty much guarantee effectively that when I go to clinic, we're not talking about how we're targeting intratumoral heterogeneity. We're not even close there yet. But is it an epigenic cause of drug resistance? Understanding how they develop heterogeneity is somewhat taken as a given, that they're vertically transmitted. Whatever is responsible for the heterogeneity is just transmitted. It's just there. Subclones now we finally get to acknowledge are just there. But what if they're not just there and they become that through communication, through links? So the T2, T3, T4 versions of what I was talking about in the translational timeline are that we know that resistance emerges when we apply standard drugs, we apply selective pressure, enriching the bad subclones, taking it back to the bench and understanding cellular and molecular mechanisms. A lot of this we just don't know. With communication as a common thread, it is also a growing field. Jim alluded to exosomes and microvesicles, whatever you want to call them. As a fellow, at the time, they were considered the cellular debris or the junk. They graduated to potentially biomarkers you can derive from the urine or the blood or bodily fluids. And now it's suddenly paving the way for the metastatic niche, the next thing that's targetable. If we hypothesize that horizontal instead of vertical cell-to-cell transfer may be responsible for propagating signals that will elicit and induce a change, whether it be transformation or making other cells more chemoresistant, is that a way to propagate intratumoral heterogeneity and intertumoral heterogeneity? That's finally getting to the part about telling nanotubes. When I went, I kind of stumbled into it by accident as a fellow, and I went to AACR annual meeting kind of in the middle of this as I was forming my very initial ideas. I went to the Nature booth and I said, wow, telling nanotubes are on the cover of Nature. This must be a cool thing to pursue. But the caption said, scanning EM of a breast cancer cell. So it was a great picture to me that these are things that are right in front of us. Molecular oncology seems to be the more sexy thing, but it was right in front of our face. It's on the cover of Nature. It's on the cover of a lot of journals, but it's not what was the attention of focus, but it has become my attention of focus. So if I show you this, I showed you this a few minutes ago, if I give you the same exact rendering with some stromal removed, it's looking at it with a different perspective. If we look at this tumor with some of the other stuff away from it, if you see a tube that's linking two cells from a human tumor, and our initial foray was into patients, about 6 patients at Memorial Sloan Kettering, would access their tumors with our pathology colleague, basically starting from a brainstorm of our what if, would it be possible to getting the tumors fresh out of the OR, taking them to the core facility, and basically staying and imaging them, and looking at what we identified as nanotube-like protrusions that were extending and connecting cells at long range between cells within the tumor. They had the characteristic of what we're seeing in vitro, and I'll show you some of those in a moment. These long, either curved or straight, very narrow, nano-width-sized protrusions. They're connecting cells, and they had the characteristic blebs that are very common, what we see in terms of the vesicles or cargo that are transported. Going back to the idea of horizontal cell-to-cell transfer. What if something could be transferred? And what if, when it goes the other side, it has an effect? It has a function that can induce chemoresistance, that can induce a cell, if it's already malignant, to become more malignant, so to speak. If it's not already malignant, can it induce malignancy, perhaps in a stromal cell? So looking at, I'm showing you again, a little repetition, but with reason. If I show you the same scratch assay, now I'm looking here, and I'm showing nanotubes forming at the invasive front. So on the battlefront of this war, in terms of strategy, now I'm seeing communication happening at the invasive front that I imagine might mimic as a 2-dimensional rendering of a 3-dimensional tumor recurrence. If that communication is occurring at the battlefront, this is a mechanism potentially for the cells to be synchronizing. They're not every man for himself along the way, but maybe they're all synchronizing as one functional unit, and it's a syncytium that goes forth and tells each other what to do. There are nanotubes in other tumors, as we look at them not just in humans, but in animal models of cancer. Here, we're looking at osteosarcoma in an orthotopic syngeneic model, and also human epithelial ovarian cancer. These are from patients with chemorefractory colorectal cancer, and as we get tumors from TPF, we did I'm trying to go back to the T2 part or so, I realized this is one of my patients. This is a patient in her mid-40s who had a very high-risk form of stage 3 colorectal cancer that was resected. She got standard of care adjuvant chemotherapy. 8 months later, she had suspicious lesions seen on her CT scan in her liver that were ultimately treated with chemotherapy and surgical resection. But this is something of interest where if we're seeing these long nanotube-like structures kind of starting to generate hypotheses that if they're present, what could they have been doing to make her tumor, to induce intratumoral heterogeneity, intertumoral heterogeneity, and to propagate and create a higher risk for her than in other cases. I was talking with Jim before the seminar about a very nice study in Nature, a group out of Heidelberg that did an optic window intravitreal microscopy over 100 days of glioblastoma, showing what they termed as tumor microtubes, and the only difference is really that they're thicker and longer, but they're essentially playing the same function. And that was what they were looking at, was interplay of angiogenesis and prognostic markers in glioblastoma. I think it was a very elegant study. So, of course, this is not, if this is a novel mechanism of long-range cell-to-cell communication, it does not exist in isolation. We think of exosomes that can propagate through diffusion from cell to cell. We can think of gap junctions marked here that are essentially connection line channels that are connecting cells in immediate proximity, but they cannot be connecting cells that are far apart. And in the mix of all that is all the stroma that I was alluding to earlier. So, in this era of social media and human-to-human communication, we can run parallels about other things that cells do: cell signaling, angiogenesis, intracellular communication, intercellular communication, tumor stroma interactions that we're recognizing may be targetable, in addition to the other things I was talking about. So the tumor microenvironment involves a lot of communication. It's necessary. So the setting for that is not just tumor heterogeneity at the molecular level, and I just use microRNAs as one example, but tumor stroma proportions that differ between patients. A higher stromal proportion has been linked to worse prognosis in a whole wide spectrum of different solid tumor malignancies. And we think of the need for long-distance communication. This is especially true in cells that undergo EMT. They get ripped apart. They have changes in self-morphology that bring them apart, and there's a higher need for long-distance communication. And if you think even just KRAS mutated cells that no longer have gap junctions and have a different morphology, there's a niche for this. So if we look at, again, I showed you this before in terms of ontology, but if we look at it in a different way, what if this was a founding clone and there are subclones here, but what if these lines are not just a phylogenetic tree, but might be actual tunneling nanotubes, or some kind of long-distance pipeline that allow the founding clone or other clones to still undergo maybe some form of mutation, but can then spread that mutation to other subclones. So if there's one we propose that's more drug resistant than another, it might propagate that and make cells more invasive, especially in the context of a patient receiving tumor-targeting drug that eventually becomes ineffective, which seems to be inevitable. So in the interest of time, I'll move forward. Some of our in vitro studies, taking it back to the bench, so to speak, it's not something that's specific to any one type of cancer, but we believe is upregulated in a whole wide range of invasive malignancies that we focus on. They are conduits for cell-to-cell transfer that's direct. So the analogy I often use is of skyways, and it's apropos in Minnesota, because if you're going through a skyway on the second floor above a street connecting two buildings, if you walk through the skyway and you keep on going, it's a very high efficiency rate, shall we say, that you'll make it to the other side. Exosomes, I don't think you can say the same thing. And the cargo can be diverse. We show microRNA transfer. We've shown Golgi apparatus. Many groups have shown mitochondrial transfer. And there's other opportunity for other cargoes that, as we take a translational approach, as I do in our lab, is think of the functional problem of specific cancers and working our way backwards. Let's think of what cargo would be of interest, what would be important or relevant clinically, and study those. So we've had 5 papers in my time here, including a forthcoming one here, specifically looking at the role of hypoxia in ovarian cancer cells. And I won't review this in great detail. I'm always happy to talk about more. But just maintaining a bird's eye view, if we look at cell-to-cell communication via nanotubes as a possible mechanism of induction of chemoresistance, we might postulate that there might be forms of cellular cargo that go from cell to cell to induce chemoresistance. So in this one study in collaboration with Subri's lab, we did analysis of differential expression between chemo-resistant or platinum-resistant and platinum-sensitive ovarian cancer cells and identified microRNAs that are differentially expressed. And then we tagged those with an Lexel floor to be able to visually monitor them going from cell to cell. So in this case, we co-cultured osteosarcoma cells one another and they form nanotubes and propagate the microRNA from cell to cell. And also in this case, looking at tumor stromal interactions with an eye towards that as well. On the right in panel B, an osteosarcoma cell will send a microRNA, an oncogenic microRNA, to an osteoblast. And so the arrow is following there, the tagged microRNA. So that opens up a what if that to me is the excitement begins now, where we can actually look at the function. What does it do when? How long does it do it for? And how is this communication taking place, and what is its impact? This is another example of an ovarian, platinum-resistant ovarian malignant cell communicating with an ovarian epithelial cell, microRNA caught in one snapshot in time from a time lapse, migrating. And that bulge, you'll see, is very characteristic. When people ask, well, some cargo you're explaining might be too small to go through a nanotube, my answer is, the analogy I give, and you can tell I'm full of analogies, is that if a python swallows a pig, it adapts, and the pig will go through to the esophagus and then eventually get digested over a long, excruciating amount of time. But remarkably, nanotubes seem to adapt to the cargo rather than the other way around. And in our paper that's currently in press at Oncotarget, one of the things I wanted to look at was, well, who's starting the conversation and who's ending the conversation and what are they talking? And we saw there was heterogeneity. There was room for that. In the first two panels, we were looking at chemo-resistant ovarian cancer to chemosensitive ovarian cancers. And you can tell here S to S is sensitive to sensitive, resistant to resistant, sensitive to resistant, resistant to sensitive, by analysis of time-lapse imaging over 24 to 48 hours, frame by frame, and looking at who starts the conversation. And so in many of these instances, the resistant to sensitive initiation of communication was not the majority, which was counter to what I might have thought. But then I'm starting to think more, based on further data, that maybe it doesn't need to be the majority of the discussion, but maybe that's enough to create chemo resistance potentially. Another common question that we're trying to encounter is, well, if tunneling nanotubes are important, they're not specific to cancer, but would they form at a higher rate in cancer? And I think it's important to keep that perspective and analyze that. The thing I also keep in mind is that what we're doing in the clinic is we're giving chemotherapy, the principle of which cancer cells proliferate at a higher rate than normal cells. They take up chemotherapy, and that's their principle. And we shed 1 billion cells or so in our colon normally every 4 days. So that's why a lot of patients also get nausea, vomiting, diarrhea, and GI side effects. Right? These are not selective, but they are targeted. But if nanotubes formed at a higher rate, if they accelerate the amount of communication, if there's a quantifiable difference, that might allow us to kind of think of them as potentially targetable structures clinically. So in this panel in particular, we looked at 3 different histologic subtypes of malignant mesothelioma, all of which carry a bad prognosis clinically in comparison to a mesothelial cell line that's benign. And there was a high ratio, anywhere between 20 to 100-fold difference in terms of the rate of nanotube formation over several days. Getting to, in terms of how they form and how they probe, if you think of the matrix in the postoperative setting, it's an inflammatory soup. It's very primordial, and it's perhaps going to make cells susceptible to grow in certain ways. The cells are scattered, and they want to communicate. And so we know about other structures that are actin-based, just like nanotubes are, but they're different in composition. And there are some called invadipodia, philopodia, and lamellipodia. In this example, I'll show you the time-lapse for this in a moment, but interestingly, there are 2 islands of mesothelioma cells. And this invadipodium sticks out and really within a few hours retracts. And in its place, you can see the morphological distinction. 3 nanotubes sprout in this place, and they have the characteristic blebs of cargo and that are transmitted to the other side. So I'll show you. Invadipodium juts out in this direction, and we think this is a probe that allows the nanotubes to sense the island of cells on the other side. And once they made that connection, they allow the propagation of the vesicles or whatever cargo. And then once the cells move towards each other with normal mechanisms of cell motility, there's no need for a nanotube anymore. So it's a temporal need that's filled by, the gap is literally filled and no longer needed. So we've looked at whether nanotubes might be a potential therapeutic target. We are in collaboration with a major pharmaceutical company that screening compounds that potentially could either inhibit or otherwise alter nanotube structure or prevent them from forming in the first place. Some of what we've done so far, in our initial publication, looked at mTOR inhibition, was successful in doing that with a number of compounds. And at Sloan Kettering, I had access to migrastatin, which is an inhibitor of FACN. FACN is an actin bundling protein, and as filaments of actin are the track, so to speak, the train track of nanotubes. They're pretty important. And then by 72 hours, they were able to reduce the number of nanotubes by targeting FASN. So it shows that they're at least targetable in vitro. But kind of thinking beyond that with my clinical cap, I go to clinic and come back to the lab and sit in front of my computer and drink some coffee and try to be mad scientist again without going too far. We think of, well, what if there are other ways that, again, cancer, knowing your enemy, knowing how to target your enemy. If I were a cancer cell and I made these conduits, why would I make them? What if we can harness them as people who want to target them rather than getting rid of them? What if drugs we're giving can redistribute through them? Or what if, said conversely, what if the tumor cells were using them for bad purposes? So in this case, the red cells are actually doxorubicin-treated cells that are platinum resistant and doxorubicin resistant. And we also co-cultured with platinum-sensitive cells. The doxorubicin is red because it's autofluorescent. But in this case, you'll see in this movie the resistant cells here with doxorubicin, and this one is sensitive, and two nanotubes form, one of which remains over time. And this is a number of hours here. But a small speck of doxorubicin is all it takes to make it into the cell through the nanotube. And then when the connection is severed, within a few hours, the cell seems to implode or undergo apoptosis. So this really is it's fuel to the fire, so to speak, in thinking, what if there are conduits that can be harnessed for drug delivery? We had the great pleasure of welcoming Dr. Yumin Fang to this Cancer Center series about 5 or 6 weeks ago. He's been a great role model of mine and collaborator for the last 5 or 6 years. We've co-authored about 5 papers and have one in which our worlds have converged. It's kind of a renaissance for me because my PhD was actually on oncolytic viral therapy, and the field was something you just went under, it seems, for about a decade, and then culminating in 2015 with the first FDA approval of an oncolytic viral vector, which is herpes, for treatment of metastatic melanoma. So we've linked together with his expertise in oncolytic viruses and ours in nanotubes to look at direct cell-to-cell transfer of, in this case, GFP-expressing oncolytic herpes simplex virus. So we wanted to think that beyond just oncolysis of cells as cell-to-cell spread, beyond just gap junction-mediated spread, could nanotubes form like this and propagate the virus, and then the virus is propagating and increasing the expression of its GFP in that case. Could it also do what's called the bystander effect? And so we have a paper currently under review on that. This is an example of a lung nanotube basically propagating the virus through the nanotube to a target cell, which then also implodes. So I'll leave you with a very nice quote that I vowed to show in every PowerPoint from now on when I discovered it. In The Hallmarks of Cancer from Bob Weinberg and Doug Hanahan. Finally, the circuit diagrams of heterotypic interactions between the multiple distinct cell types that assemble and collaborate to produce different forms and progressively malignant stages of cancer are currently rudimentary. In another decade, we anticipate that signaling circuitry describing the intercommunication between these various cells within tumors will be charted in far greater detail and clarity, eclipsing our current knowledge. So finally, in the words of Sun Tzu, in the practical art of war, the best thing of all is to take the enemy's country whole and intact. To shatter and destroy is not so good. So what I'm looking to not shatter and not destroy as a human patient, as we're targeting their tumors, we also have to keep in mind what strategies and tactics can I use to go in for the targeted kill, but enrich the life of the patient and not destroy them in the process. So too, it is better to recapture an army entire than to destroy it, to capture a regiment, a detachment, or a company entire than to destroy them. So my team is really grateful for my team. So Venu, Phil, Snyder, Liz, and Justin basically did all the nanotube work. They help out a lot with the other studies as well. I have a really great team of mentors and collaborators. Cliff Steer, Greg Versalati, and Dick King have been great role model physician scientists. Subri Subramanian, Al Exxon from engineering. When they say it takes a village, for other things, I think it takes a village to do translational science as well. Melissa, Deanna, the pathology team, the biostatistician. When you talk about languages and being able to, I can interpret certain languages in science or outside of science, but when it comes to biostatistician and genomic sequencing, I don't pretend to know these languages, but recruiting people into your team is essential. I'm grateful these people have joined our team. And outside the institution, it's important to really talk with people who are like-minded and maintain collaborations as well. I'm grateful for folks who have supported the work monetarily also, and I'm happy to end it there and welcome questions.
So one of the things we talked about was, we talked about the microenvironment, and in addition to the different cell compositions and matrix, there's also physical changes, or like hypoxia, for example. And do those sorts of regional changes in the tumor, do you see a difference in the propensity of these structures when that... Yes. You see a propensity of those structures in that type of environment?
Definitely. So, the things that I didn't get into in terms of our pure data on it, but these nanotubes are elicited in cancer or magnified in times of metabolic or physiologic stress. So our paper that's in press, timely enough, has investigated the role of hypoxia. So we know it's a very relevant tumor microenvironment process that impacts the way we treat tumors, induction of angiogenesis, which is a hard thing to overcome clinically. So in our studies, we've actually looked at differences in nanotube formation between cells that basically looked at nanotube formation in cells that were exposed to hypoxia versus normoxia. We also just verified that HIF-1 alpha as a marker of hypoxia was upregulated in our cell lines. We looked at chemoresistant and chemosensitive cells and did find that the chemoresistant cells, SCO3 and C200 are platinum-resistant forms of ovarian cancer. And A2780 is a parent line of C200, chemosensitive cell line. Basically, the overall finding was that the cells that are chemoresistant had a higher propensity for forming nanotubes in a hypoxic condition, but the chemosensitive cells were not. So that leads us to believe that...
Sure, absolutely. So if you have follow-up to that, have you been able to shut down HIF-1 alpha and prevent their formation? Have you done those experiments?
No, that's a great concept. I'll also add that HIF-1 alpha itself can be transferred between cells via telling nanotubes. And so one of our proposals currently is investigating, basically pursuing this line of work and findings. To look at the role of hypoxia and HIF-1 alpha and what might be able to be targetable or knocked down to knock down nanotubes indirectly, and whether or not knocking down those nanotubes would prevent the cells from being as resistant.
Are there other questions?
Question there.
Yeah.
Host cell. And so if you think about it like that, you might expect them to be covered in a similar milieu of receptors as the host cell membrane. But is that necessarily the case? Are there certain cell membrane proteins that are enriched or depleted along the length of the nanotube?
Yeah, it's a great question. I think that one of the holy grails is or the golden ticket or whatever you want to call it, is identify a marker that might be nanotube specific. So the whole field is in its infancy, and some of the things that we've looked at that we have identified are lipid raft enrichment. Of course, lipid rafts are not specific to cancer cells or nanotubes, of course, but they're enriched several fold in cells that are forming nanotubes as opposed to not. Some groups have looked at in non-cancer cells, integrins or cadherins and other markers that you're right are, as we think of them, they're an extension of the plasma membrane. So it would stand to reason that whatever's on the plasma membrane of the cell body would also be on the exterior of the nanotube. So again, novel approaches of thinking of it and perspectives are, should we think of and identify or pursue a target that's on the membrane portion of the nanotube, or can you shut it down from the inside, like something like Jim was mentioning, or both, multi-pronged attack as ongoing investigation.
So is the lipid raft formation the reason the mTOR inhibitors affect the nanotubules, or is mTOR inhibition a different mechanism?
That's a great question. We examined them separately in separate studies, but that's a good point. I didn't go into detail, but one of the citations I showed, I also think, again, with the perspective on this role of exosomes and microvesicles has really skyrocketed exponentially in the last few years. I don't see them as mutually exclusive. So in one of our studies, we actually isolated exosomes derived from tumors and added them to cells, and then the cells ended up forming more nanotubes. But if you took exosomes derived from benign cells, they had no effect. So not only would they stimulate more nanotubes, but they would actually shuttle through the nanotubes. So in my mind, I'm like, I think it makes a lot of sense. So maybe the cargo doesn't have to go naked through. Maybe it's protected, at that point, whatever you want to call it, exosome or endosome, depending on its location. But in essence, it makes a whole lot more sense to me for an exosome to want to be protected and shielded and go through a very efficient conduit, skyway-like conduit, than to just freely diffuse and have some of them make it to the target cell.
Are these nanotubes present in all cancer types?
So I alluded to one slide where we focused on invasive cancers, and so we know of at least 8 or 9. Again, looking at invasive cancer types. There was a nice paper in Blood that appeared in late 2015 looking at CML. So I think it's from my view, it's a universal process, but I think there's something different about the nanotubes in cancer cells, whether it be just purely quantifiably, there are more of them, or if they have different characteristics, property-wise and structurally and functionally in cancer is still something we're investigating. Yeah, sure.
So have you looked at other signaling pathways like the small GTPases? Do they have to be active in order for these things to form? You'd think they would be.
Right.
And then the second thing would be like transmembrane tyrosine kinases. A lot of times those will stimulate motility in addition to growth. So, and I guess one more. Is it cell cycle specific? Is there something in the stage of the cell cycle where they form?
Right. Right. No, those are all excellent points that we've had to address. So working my way backwards. So one of the things that we try to do is also examine cell proliferation in tandem with a number of nanotubes to ensure it's not purely a function of there are more cells, therefore proportionally more nanotubes. The cell cycle thing is something that's been on our plate, but looking at cell cycle inhibitors, one of the challenges has been I don't know that we'll find a drug that will only shut down nanotubes and not have any impact on cell cycle or cell proliferation.
Oh, absolutely. But I mean, even synchronizing cells and then tracking them through cycle to see at what point do these form.
Correct. Yeah, and that's definitely been on our plate and it's in a proposed set of studies.
Thank you so much.
Thank you. Thank you.